is, let's focus on feeding pathways. Right, feeding pathways. So in feeding pathways, so again, our objective is what? What is our objective? We just talked about, right? Objective is in the blood vessel, in plasma, there is 100 grams per deciliter. But if it's more than 100, like let's say you just ate, it's 500 or 400, whatever that may be, right? Your goal is to remove that. So remove excess amount of glucose from plasma, right?
Mm-hmm.
So why is that objective? Why do you want to do that? Why? Because if you don't, so there will be cells here, right? Let's say this is a cell here, this is a cell here, this is a cell here. So because sugar, glucose is osmotic molecule, right? So if you have excess amount, 500, 400, 600, whatever that may be, right? It's going to pull all this water into the blood vessel, right?
Mm.
Right. So from the cell, so you have shifts of intracellular fluid into ECF, which is extracellular, right? So in the ECF, you have sodium sitting here. It's like usually like 135 to 145 is normal, right?
Right.
And that is given that you have 100 grams of glucose levels, right? That is normal. But if you have excess amount of sugars that are present in the blood, and now you're pulling all this water in, right, from the cells. So what are you doing to the, because of this osmotic pressure, it's going to pull all this water back into the vessels. So what are you doing to these cells, right? So what are you actually doing to these cells is, let me just copy this here, copy and paste here, right? So what are you doing to these cells is, you're shrinking them. Right? So this is what happens. So originally your cells are like this, right? So your cells are kind of like this, and now they're shrinking, right? Because you sucked all the water into the blood vessels, right?
Yeah.
So what do you do to you? So since this was your normal sodium, now you have lots of water, so this will dilute. So now you have, like, let's say maybe you had a 10 liters before or something. Now you have a 20 liters here or whatever. So what is the molarity is equal to? Molarity is equal to mass, which is solutes, over liters, right? So what you just do here is you pulled a lot of water, so your denominator is going up, and your numerator is staying same, right?
Right.
So what do you do to your sodium? You dilute. So this is what we call is dilutional hyponatremia, right? This is why most of these patients, who have excess amount of glucose, they are not able to remove, such as in DKA and HHS. They will not be able to. Their sodium is diluted, which means it's falsely diluted, which means their sodium levels are normal, but because of the excess amount of water that is present, that is why their sodium is diluted, right?
Yeah.
So this is why, to prevent, when these neurons going to shrink, right? So this can actually cause altered mental status. Right.
Mm.
So this is why you have to be really careful. So our body, so what does our body maintains homeostasis, right? Through hormonal mediated pathways, right? So when these pathways, when these hormones are messed up, when these hormones are not present, or there are receptors down or up regulation, these pathways won't work. won't work how it's supposed to, right? So today, what we'll focus on how does our body maintain this homeostasis through the normal pathways, right? And then we'll talk about, what happens when these pathways are not working.
So first, we're going to talk about how a normal human being functions on a daily basis,
right?
And the problems that could arise from it.
And then we will talk about this. And this is where we'll focus on DKA, HHS, and all of that stuff, right? So in order to understand this in proper detail, like why we do the things we do in DKA, we need to understand what is happening at normal,
right?
Like normal physiology of these hormones, right? So then let's go into feeding pathways, right? So our objective is remove excess amount of glucose in plasma. How does your body remove excess amount is what we'll talk about, right? So feeding pathways again, right? So how does our body does that? So this is again blood vessel. There's a lot of excess amount of glucose here, right? So the pathways that I want you to really think about whenever you eat from next time, anytime you eat, I want you to focus on these pathways, seriously.
Okay.
So first one is glycolysis, right? So that I want you to think about like burning glucose. Okay? Glucose, glucose, and lysis means breaking it down, right?
Yeah.
So what else can you do? So, well, you can actually break it down. First, in order to break it down, you first need to remove this, right? You need to remove this. So the most important organs that actually helps you remove this, you need to know that is the liver. Liver removes glucose at high concentration. When substrate concentration is high, which is substrate here is glucose, right?
Mm-hmm.
Our liver has an enzyme called glucokinase. Glucokinase works very efficiently at high substrate concentration. You need to memorize that. Okay. So at normal concentration, like if you have, let's say, 100 grams, this glucokinase is not working as much.
Okay.
Right? So what happens is, this is the curve, right? And then this is let's say, if the Km is high, like it has lower affinity, which means it's gonna be able to burn a lot more glucose, trap a lot more glucose into the liver by glucokinase enzyme when the concentration is high, when the Km is higher.
Gotcha.
Right. So in this case, liver does that. Okay. So liver will take this excess amount of glucose, and then other cells that actually does that is your fat cells, adipose, right? Skeletal muscle, and also cardiac muscle. So how does these cells actually remove this glucose? Like a heart here, there's skeletal muscle here, right? And there's adipose, right? So that's also removing glucose, right? But these cells don't have glucokinase. So they need transporters here. These transporters are called GLUT4 transporters. And in order for these transporters to translocate on the membrane, you need insulin. Okay? So you need insulin for those. And then this one is, the liver doesn't need GLUT4 transporters, but it has an enzyme that is only going to work, trap all this, this is the liver right here, right? And it's going to trap all this glucose into the hepatocytes by glucokinase, which is only going to work at low concentration or high concentration.
High concentration.
Right. So that's why it's this is how our body works, right? So this enzyme only works at high concentration. Okay. So one is you can burn. So once you uptake these glucose right into these cells, right, and then you already start removing the glucose from your plasma in the liver, in the heart cells, and in the skeletal muscles and adipose cells, right? So then once the glucose in the cells, right, then you can do glycolysis, number one, right? What else can you do? You can do you can store the glucose into these cells, right? And then when you store, what do you call that? Storing glucose.
Glycogenesis.
Glycogenesis, right? So basically you're storing in the form of glycogen. What else can you do? Well, you are doing glycolysis, you're burning glucose. What is the end product of, so let's say this is the glucose. Now we're focusing on the cell, right? So let's say this is one of the cells here, right? You trap the glucose. How do you trap the glucose? Well, you have an enzyme called hexokinases, glucokinases, remember?
Mm-hmm.
So hexokinases or glucokinases in the liver, hexokinases in these cells right here, right? So what they will do is they will trap these, glucose, and then by putting a phosphate on it, right? So this will be glucose 6-phosphate, right? So now you're trapped, right? So now it cannot leave, right? So what happens is then this will go through certain pathways, and then you will get your end product called pyruvate, right? This is all right. So you're burning glucose, that's number one, right? So this is your number one pathway, right? And then you're storing glucose, so you can store some of this here, number two, in the form of glycogen, right? And then you can also, you still have a lot of glucose to burn, right? So what you will do is you will shift this pyruvate into mitochondria, and then you will keep on, convert this into acetyl-CoA, and then you keep on, go to oxaloacetate. Can I keep selling S for money? Oxaloacetate, right? So remember those, Krebs cycle pathways?
Oh, yeah.
Yeah, so you will keep on making, what happens is your ATP and NADH goes up. So this will, basically you're filling up your ATP stores, right? And that would be Krebs cycle. Make sense?
Yeah.
So you got this Krebs cycle. So let's say you got your glucose, you're burning down, making pyruvate, and you got your glycogen. You fill these stores because you can only store so much glycogen, right? And then you got your pyruvate, and then you still need to fill up your ATP stores, so you need to go to mitochondria, and this go to mitochondria right here, right? And then now you're filling up your ATP stores. Your ATP stores are filled, and now what can you do is, when your ATP stores are filled, this citrate right here will come out of the mitochondria. The citrate will come out of the mitochondria. So let me just, erase this here. Right? So now you're back in the cytoplasm, right? And this citrate will be breaking back to acetyl-CoA, right? So this is stops, and now citrate is, because you filled up your ATP stores, now citrate is accumulating, and it's coming out of mitochondria by something called citrate shuttle, right? And then you keep on making, a lot of citrate, right? Because you still have a lot of glucose you're removing, right? You still have a lot of glucose you're removing, right? In the cells, you're burning the glucose down to pyruvate. You made your glycogen, these stores are filled. You can store that into the liver. You can either store that into the skeletal muscles. And now on the top of that, you're still burning down pyruvate. And then you can fill up your ATP stores. And this is all happening, by the way, in the feeding pathways, right? When you're eating, you just ate within the last 10, 15 minutes, right? and then the pyruvate is going into the mitochondria, and then you filled up your ATP stores, and after you filled up your ATP store, the citrates start going out, right? And from there, you have your acetyl-CoA, and now you have acetyl-CoA. Once you have a lot of acetyl-CoA accumulating within the cytoplasm, and given that you have you're in the feeding state, this acetyl-CoA can be used to make two things, right? So what are those two things? One is palmitate, which is fatty acids, right?
Mm-hmm.
What is another one? So we'll actually expand this further a little bit. So palmitate, and what's what else can you make from here? Cholesterol. Okay, so when you make palmitate and cholesterol, and these can be combined in VLDL. So these are your VLDLs, right? And these VLDLs pack your palmitate and cholesterol. So palmitate, when 3 palmitate combines, will make your tags, and plus you forming those cholesterols here, right? And so these VLDLs now go into the blood, right? And they get shifted to the fat cells, right? These fat cells right here, let's say there's a fat cell here, adipose cells right here. And they have something called lipoprotein lipases on those vessels. So they will start cleaving these triglycerides into these and then keep on storing it. Okay. So that's pretty much happening in the feeding pathway. And that is why when you want to draw blood levels, right? When do you want to check lipid levels? Like right after the eating, or they should be fasting?
No, they should be fasting. That's something that we always do in the hospital.
Okay, so why though? Because during feeding, you just falsely elevated, VLDLs, right? There's so much increase, so when you're actually measuring their blood levels, they will have like way more higher VLDLs than normal, right? So that's why we don't want to do it. So like these will be elevated when somebody just ate, right? So if somebody just ate and then you're measuring the VLDLs, it will give you false elevated, like hyperlipidemia.
A false reading, yeah.
So like even like if the person is like, let's say 20 years old, they just ate like a big burger, right? And then they are going through these feeding pathways, and they will have a lot of fatty acid synthesis going on during feeding. And even though they're exercising and everything, if you measure their blood levels, right after they ate, their VLDLs, LDLs and everything else will be elevated.
It'll all be falsely elevated.
Yeah, exactly. So for that reason, we do it during fasting, right? So that is feeding, right? So then this is what we do, right? So again, number 4th one was fatty acid synthesis, right? And what was number 5th? Number 5th was cholesterol synthesis, right? So if you want to start somebody on medications for cholesterol synthesis, you better not be measuring their VLDL and cholesterol levels, right? During just, somebody just ate 1 hour before they draw their labs. You shouldn't, technically they will be in fasting state, but you should just fast at least for a good 8 hours.
Yeah, good 8 hours beforehand. Yeah. Right. So that's why we always say skip breakfast when you go and get these labs done.
Exactly. So somebody just, went to sleep at 12, they should be able to draw their labs at 8 and not have breakfast and get them breakfast afterwards.
Yeah, perfect.
Right. So then is a fasting, right? So one thing I forgot to mention here is which hormone is elevated in feeding. So again, why do we need insulin? Because so in feeding, you will have a lot of insulin increase. Okay. So you have a lot of insulin increase to do what? So one is to put this GLUT4 transporters on these cells, right? That is one. What is another one? Another one is all these pathways here, like these pathways, like this pathway, these pathways, and even this, and even this. Some of these pathways, most of these are upregulated. Most of the enzymes in these pathways upregulated by insulin. Right? So if you don't have insulin, you will not be able to do a lot of these things, right?
Yeah.
So which means you will not be able to reduce your blood plasma glucose levels, right? So if you don't have insulin, you will not be able to remove this glucose into these cells, right? So which means your cells will be not able to make energy and all of that stuff, right? So this is really, really important, right? So these GLUT4s are upregulated by insulin. What is number 3? Well, we talked about you need GLUT4 transporters, right, to remove this. What about liver? Liver does liver need insulin?
No.
It actually does need insulin. How? Because you're going to upregulate glucokinase. Insulin, upregulate glucokinase, right? Kind of makes sense, right? Because if you don't have glucokinase, for example, if you do not have a glucokinase, will you be able to remove excess amount of this glucose from the blood?
No.
So that's why you have insulin that actually is going to work on glucokinase. It's going to basically increase the synthesis of glucokinase. It makes sure when you're eating, your glucokinase is present in higher amount. So that way, as your concentration of glucose goes up, this enzyme is working, the Vmax is actually going higher and higher, which means like you're breaking down more, you're trapping more of this glucose in the form of glucose 6-phosphate, right? This is really, really important, right? So glucokinase is present in the liver. It's going to basically, once insulin goes up. And there's another mechanism, right, why glucokinase is also present in the beta cells of the pancreas, which actually makes the insulin. So that's another mechanism which we'll discuss later. But for now, I want you to just focus on insulin upregulates glucokinase. So if you have a glucokinase upregulated, you're going to remove all this excess amount of glucose in the liver, and you're going to do all of these things. And also, you have GLUT4 transporters that are translocated, and now you have these transporters present on the plasma membranes of these cells, right? And now you're shifting a lot more glucose into these cells, so you're burning it down, right? So that is once these cells have glucose, what are you doing with those? You're burning it down, right? For example, your liver is burning it down. Your heart is needing a lot of ATP, right? It's going to burn it down.
Yeah.
And you're storing glucose, and you store in two main organs, right? Glycogen. Glycogen can be stored in many places, but like the most one I want you to know is in liver and skeletal muscle. Okay? And then you stored it, and then you still have a lot more pyruvate left, right? And then what are you going to do is you're going to shift some of this pyruvate into the Krebs cycle to make a lot more ATP, right? So once you make a lot more ATP, then your stores are filled, and your body's still want to burn a lot more glucose. It's going to stop this this TCA cycle, because your ATP and NADH accumulates in higher amounts, right? And then this will start shifting the citrate into the cytoplasm through the citrate shuttle, and now your citrate is here. Now it's converted back to acetyl-CoA, right? And then acetyl-CoA is used to make fatty acids, which is palmitate, and it's also used to make cholesterol. And these palmitates and these cholesterols are packed into VLDLs and sent out, and then these VLDLs are traveling to the fat cells, and the fat cells have something called lipoprotein lipase. So interestingly, on fat cells, these lipoprotein lipases are also upregulated by which hormone? Insulin, right? So everything is working in very coordinated fashion, right? So that is why insulin is so important. And that's why if you don't have insulin, these people will have a lot more hyperglycemia, right? And that's why they will have a lot more VLDLs, which means these VLDLs, a lot more tags, right? So in DKA people, in diabetes people, they have a lot more tags, right? Because they have less lipoprotein lipases that are working, right? So that's why these are upregulated by insulin. So you have to kind of understand the normal physiology. So then we go into